All right. So we've said that the job of the plasma membrane is to keep stuff out, to keep stuff in the cell, and to let the good stuff pass between the cell. Okay. And that's what we're going to talk about with this transport. And remember, when we looked back at that concept map, we said that we're going to talk about passive, we're going to talk about active, and we're going to talk about bulk transport. Okay, so here we are, we're going to spend this section talking about passive transport. Okay, the defining feature of passive transport is that it does not use ATP, right? No energy is needed. So once we get into that, we can talk about simple diffusion and facilitated diffusion, both of which are going to be examples. Simple, facilitated. Okay, those are both examples of passive transport. We'll look at the types of proteins, specifically channel proteins and carrier proteins that allow simple and passive transport to happen. And then we'll look at a special type of passive transport called osmosis, which usually scares people, but not you, because you are a big smarty pants. Okay, so let's define some terms. So plasma membranes are what we call semi-permeable. This means that there's going to be selective permeability, that some stuff can pass through the plasma membrane and some stuff cannot. Okay, so you make an analogy. Here's my cute doggy Maggie. She's the cutest little pie. This dog is really good at finding forest bones. So she will find a bone in the forest and she will carry that bone back for 2 miles back to the house. Now, forest bones are not allowed to enter my house, but grocery store bones can. So my front door is acting as kind of the bouncer. Forest bones are not allowed. Grocery store bones, come on in. Okay. My front door, in this case, is semi-permeable. Some things are allowed to come through, some things are kept out. Okay. There are also some things that the dogs are not allowed to take outside. Like my good kitchen towels that they steal off the stove and try to take them outside because they are really good at things to play tug of war with your doggy sisters. Okay. All right. So our plasma membrane is semi-permeable. Some stuff's in, some stuff's out. So the things that can pass through and things that can't pass through are going to be determined by their properties. Okay. So small particles, whether this is going to be, so I'm going to write particles here. Ions, I mean molecules, right? So it's kind of a big blanket term. Things that tend to have high permeability are small molecules. They tend to be uncharged. They just kind of wiggle its way through the spaces between the plasma membranes. Okay. It's important that these things are uncharged because they have to pass through this fatty acid layer, which is, as we know, hydrophobic. So in order to pass through that, you also have to be hydrophobic. Okay, if you're a charged particle, and you can hang out with these phosphate groups, but once you get to those fatty acids, they're like, nope, you get out of here. We don't like you. Some other things that have low permeability are large things. So amino acids, they're really big, really complicated. ATP, it's big and charged. Proteins are usually really big. Polysaccharides are freaking huge. Nucleic acids, we don't want those escaping. They're large and charged. You do not have to memorize like all of these things, these examples of this whole spectrum of permeability. But what I do want you to know are the ends. So okay. What you need to know is in black on this slide. We need to know that there's a spectrum of permeability through our plasma membrane. I do want you to know the characteristics. I want you to know the characteristics of things that are highly permeable. And I want you to know the characteristics of the things that are at low permeability. And then I want you to be able to apply that. So if on an exam question, if I said, I gave you the chemical structure of atmospheric nitrogen, and I would even remind you that this is a non-polar bond. And you are going to know that it's a small molecule. You would go, aha, yes. If it's a non-polar small molecule, whoop, it probably has high permeability. It can sneak its way through those phospholipids. Those non-polar bonds are going to allow it to pass through the fatty acid tails of the phospholipid bilayer. If I said, I might use hydrogen ions as an example. It's small, but the reason that it's not permeable is because it is charged. Okay. So make sure that you understand that. If you have questions, throw those in that discussion board. Email me. I'm happy to get some of those questions answered and cleared up for you. Okay. So I like this little cartoon that shows the difference between active transport and passive transport. So active transport is like riding your bike uphill. Oof, it has a lot of energy input. Okay. If you are, once you get to the top of the hill, then you get to be like, woohoo, now we're going to ride downhill. When you're running downhill, in order for your bike to be propelled forward, you do not have to have energy input to move your bike. Gravity is going to help you with that. Sometimes gravity helps you too much and you crash on your mountain bike, and then that really sucks, and then you're sad. No, that's not a highly specific example about what happened to me. Because we're talking about cells, when we're talking about energy, we are going to be using this thing called ATP. But when we're using passive transport, we don't have to use ATP. Diffusion is a really important process. So what diffusion is, this is the net movement of particles. Again, remember, these can be ions, or these can be molecules. They could even just be an atom all by its little selfie. Where we're going to go from an area of high concentration to an area of low concentration. Okay. And what we're going to say is that that is going to be going down to the concentration gradient. So if we kind of think of it like this little slope where we have a high concentration down to a low concentration. Solution is composed of a solvent with solutes dissolved in that solvent. Okay. So if we're using Kool-Aid, the water is the solvent. Our solutes, if we're making Kool-Aid, is the Kool-Aid powder. and the sugar. Okay, and our solution is Kool-Aid. Okay, so when we're making Kool-Aid and we first dump our Kool-Aid powder and our sugar in, all of those solutes are going to be bunched up in a very tight arrangement, right? They're going to be in a high concentration. And these square brackets that I'm drawing in now, that's the lazy way that we write concentration without having to spell that big crazy word. Okay, so in this case, we have a high concentration of solutes. Now, over time, due to just random movement, so this solute's going to be like wiggling around, it's going to bump into that guy, and then that guy's going to get like billiard ball pushed over there. And then they're going to be bonking into each other and ricocheting off until eventually they're due to just random movements. We're going to reach equilibrium where the space between each of the solutes is going to be maximized. Okay, so eventually we're going to have a lower concentration of our solutes just through the random movement. Okay, so this high concentration is at a high energy state. And that's where the solutes are spread out. It's at a lower energy state. This again is a spontaneous process where there's no added energy needed to go from this high concentration, high energy state to this low concentration, low energy state. And that is the mechanism that passive diffusion relies on to reach equilibrium. Okay. Because we're depending on we're depending on diffusion, we do not have to add ATP for this to happen. Okay, so let's talk about two different flavors. So first we're going to talk about simple diffusion, which is where we have our solutes are just going to slip between the phosphates to get from the outside of the cell into the inside of the cell. This is going to be driven by the random movements of those particles. Okay, because this in this drawing we have more purple circles on the outside. This is our high concentration area. Here's our low concentration area. Once we reach equal concentrations on the outside and the inside, then the net movement will stop, will reach equilibrium when the concentrations are equal. Okay. All right, let's talk about facilitated diffusion. This is our second type of passive diffusion. This is where we're going to see movement of solutes. In here, this time we're going to talk about a little blue solute that's moving down its concentration gradient. So we're going from an area of high concentration on the outside of the cell to low concentration, and which happens to be on the inside of the cell, in this case. Okay, but the difference is that these solutes require a facilitator. Okay, this protein is going to facilitate. I always run to the room. It's going to facilitate the movement of the solute. Okay. So let's think back to the types of particles that are able to do passive, or to do simple diffusion. What kinds of particles can do simple diffusion? Okay, in order for a particle to pass through those fatty acids, they have to be nonpolar. And they have to be neutrally charged, right? No charge. And they have to be small enough to slip through. So some examples could be our gases. But if you are a charged particle, or you are a large amino acid, or you are a glucose molecule, you're charged, you're polar, you're too big to get through these, you're going to get boinged off. So you need a super deduper visor. Okay, so this protein is going to then be coated in this little channel here with amino acids that are going to be hydrophilic, so that our little hydrogens can pass through. Or it's going to allow a large enough passageway for our large and polar molecule of glucose to actually get into the cell. Again, if we're looking at the outside of the cell, our extracellular fluid, so that just means it's outside, and then our gooey inside, so inside the cell, we can have ion channels as our facilitated diffuser. So typically what we see is that these ions, right, these are going to be charged particles. Let's just put little pluses on there just for funsies. They are going to allow that charged particle to get into the cell. We also see, right, so this is going from high concentration of these things to low concentration of these things. In this case, what we're seeing here is that on the inside of the cell, we have high concentration. And so the ion channel is going to allow the passive diffusion of these charged particles down their concentration gradient, which in this case happens to be going outside of the cell. Okay, so it's all driven by the concentration. Okay. Where is that concentration gradient? Okay, a cool example of this are these special things called aquaporons, which is ion channels for the movement of water molecules. And these are really important for kidney function. So depending on if you have been dehydrated, your body's going to want to hang on to the water. But if you've been drinking a ton of water, it's going to get rid of it. You'll learn a lot more about that if you take anatomy and physiology. We can also have gated channel proteins. So in a gated channel protein, there has to be a stimulus that is going to allow, so we see this channel, but this solute is too large to fit through there. So just like at your parking garage, when you drive your car up to it, and you pay your little, you scan your little ticket and you pay your money, the gate will open up, right? That's the stimulus, so that you can drive your car through. So in our gated channel proteins, we're going to have it's the parking garage situation. Okay, so the solute is going to come and dock and land in this little pocket in that three-dimensional shape of that protein. That is going to have the stimulus to have the protein change shape and allowing that solute to then enter the cell or to move across the membrane, I should say. Again, we're going from our high concentration, in this case, shoop through to our low concentration. Sometimes we're going to see facilitated diffusion be done by these carrier proteins. Carrier proteins are really specific, meaning that they are going to only transport certain solutes. So they'll only bind with certain stuff. So what a carrier protein is going to be doing is we're going to have this solute come and land in that pocket. This time this channel is completely closed. So once we have that landing in that pocket, we are going to then change the three-dimensional shape of that carrier protein, release the solute into the cell or out of the cell, depending on the concentration gradient. And then once that protein releases the solute, then it's going to go change its shape back to the original shape. And so it's going to have this cycle. What we see with carrier proteins, importantly, carrier proteins can be used in both passive transport and active transport. So they might require ATP depending on the situation. Okay. Again, looking at these, this should say carrier proteins. So let's look at these three different types of carrier proteins, where we might have a SIM port. So SIM means same, where we are going to have two different solutes. Okay, so I'm gonna make this more colorblind friendly. Okay, so we have two different types. We have the X's and O's, or red and green. So we have two different solutes that are moving in the same direction. We might have a uniport. Uni means one. Where we have one solute moving through, because we only have one solute. There can only be one direction. And we might have our third type, is called an antiport. So this means against, anti. So we're having two solutes moving in different directions.